Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Nofar Azriza binti Azman. Today I would like to present about my final year project with title Environmental Fate of Macrolide Antibiotic Degradation Pathway. Macrolide antibiotics are classified according to the size of the macrocyclic lactone ring, which can be 12, 14, 15 or 16 membered ring macrolides. Macrolide prevent protein synthesis by attaching to the bacterial ribosome, preventing enzyme peptide transferase and blocking the translation of messenger RNA. Macrolides are large-size, broad-spectrum antibiotics used to treat various infections, including skin, respiratory, digestive, and genital tract infections. One segment in this review is macrolide antibiotics are a common developing contaminant found in environmental media. When they enter the environment, they can interfere with organism growth, development, and reproduction. Antibiotics are essential in the treatment of infectious disease. However, the increasing release of antibiotics in water and soils pose a risk to all microorganisms. The objective of this review is to investigate the degradation pathway of macrolide antibiotic in environment. Antibiotic resistance is caused by alteration in a bacterium genome and foreign DNA introduction. Antibiotic may harm the environment, hastening the development and spread of resistance. This substance enter the environment through human and animal excretions, poor disposal of leftover medications, and pharmaceutical industry waste. Antibiotic entering aquatic habitat due to human pressure can impact microorganism communities, alter their architecture, and influence drug-resistant mechanism. This pollution is harmful to organisms, and antibiotic has been discovered detrimental to amphibians. Antibiotic can impair development and behavior, potentially impacting animal populations even after they are purged from the environment. The microbial population in sewage system may be impacted by macrolide antibiotics. Antibiotics disrupt animal microbiomes, leading to dysbiosis, which can cause health consequences like developmental defects, allergies, metabolic diseases, and increased susceptibility to pathogens in higher organisms. There are 4 enzymes that help in degradation of antibiotics. Macrolide esterase exploit macrolide function, relining macrolides and blocking protein synthesis. Macrolide phosphotransferase are enzymes encoding MPH genes that phosphorylate macrolides in a GTP-dependent manner, thus inactive them. In B-lactamase, B-lactam ring of antibiotic and inhibit penicillin binding protein and develop resistance to antibiotic. Lactase, versatile enzymes from various species are essential for treating and enhancing water quality by eliminating pollutants. Chemical oxidation macrolide antibiotic is degraded through free radical produced by a chemical reaction that react directly with the antibiotic, causing its chemical bond to break or decompose. In photocatalytic degradation, it utilizes light energy to drive antibiotic degradation by oxidizing and hydrolyzing through absorption in visible, ultraviolet, and infrared regions. This leads to photo-induced electron and hole pairs which react with oxygen, water, and hydroxyl groups. In biodegradation, use of microorganisms, microbe, and enzymes to break down the antibiotic in the environment. In 2002, according to Susala, plants take up antibiotic from river water and bottom sediments through roots, stems, and leaves, and then transport them through transpiration or degradation by microorganisms enriched by the roots, thus reducing content of macrolides in the aqueous environment. The degradation of macrolide in soil environment can be done by some methods which are advanced oxidative process. This process is rely on in situ chemical oxidation. Oxygenants are added to contaminated soil and groundwater, causing a chemical reaction to destroy contaminant and produce harmless byproduct. These oxidants are injected underground into wells, reaching both dissolved and undissolved contaminants, and then spreading to surrounding soil and groundwater. Ozonation. Ozone primarily attack the double bond, aromatic ring, and amine functional group of macrolide antibiotic in an aqueous form. Ozone react via direct 1,3 dipolar cycloaddition to C double bond C, C double bond N, and N double bond N and in situ generation of hydroxy radical. Photophantom reaction molecule transition from steady to excited state by absorbing electromagnetic radiation, completing degradation. Photocatalyst absorb photons, generating electrons and holes. Oxygen and water molecule interact, forming free radical and hydroxy radical. UV light irradiation reduce and regenerate the reaction system, thus producing ferrous ions and ion. Lastly, microbial degradation. In another study, biodegradation of macrolide is by removing the metal functional group. Once the hydrolysis opened the ring, the carbonyl group was removed, followed by removing the amine group that leads to successive removal for further 2 methyl and 3 hydroxy groups. In conclusion, antibiotics are emerging contaminants found in various environmental compartments. Excessive antibiotic use may harm the environment, thus removal and degradation of antibiotic can be done by several methods that I have mentioned before. In future, researchers may grab a chance to develop efficient and low-cost treatment technology for controlling the emission of macrolides antibiotic contaminants. Moreover, antibiotic-contaminated agricultural products may impact human health, thus attention should be given to indirect effect of drinking water and food chain.